I think we're live. So, hello everyone. I'd like to welcome you all to Dr. Nadine Gogolla's Worldwide Neuroscience Addiction Seminar. My name is David Haggerty. I'm a third-year PhD student in the laboratory of Dr. Brady Atwood at Indiana University School of Medicine. And my co-host, Dr. Eric Nestler, I think is here in the chat, but not on video. But we'll see if we can fix that for the Q&A session. And I'm going to go ahead and introduce Dr. Gogolla. Dr. Nadine Gogolla is currently a research group leader at Max Planck Institute of Neurobiology. Dr. Gogolla conducted her undergraduate studies at the University of Marburg in Germany before completing a master's degree at Insulin in Paris, France, in the laboratory of Dr. Roland Lebau. Dr. Gogolla then began her PhD in neurobiology at the Friedrich Miescher Institute of Biomedical Research at the University of Basel in Switzerland. Under the mentorship of Dr. Pico Caroni, she explored the regulatory mechanisms of neuronal plasticity. After finishing her PhD, she crossed the Atlantic for a postdoctoral fellowship at Harvard University under the mentorship of Takao Hensch. During her postdoctoral fellowship, she investigated inhibitory neurocircuits in mouse models of autism spectrum disorders and began to describe key developmental changes in the insular cortex in these models. In 2014, Dr. Gogolla returned to Germany and continued her work on the insular cortex, where she currently uses in vivo two-photon.
So, okay, I hope you all hear me. I will start sharing my slide in a minute as soon as somebody confirms you can hear me. Okay, great. Then I will do this now. All right. And somebody has to shout in if you can't hear or see what I'm saying. Okay. Hello, everyone. This is a little bit unconventional platform for me, so sorry if there were any glitches, but I hope you can hear me. It's my great pleasure to give today's seminar on our research, which is on keeping the balance, and we describe a role for the insular cortex in emotion homeostasis. So in my lab, we're really interested in trying to understand how emotions are laid down in the brain and how what are the mechanisms giving rise to them. And so I brought to you these pictures to illustrate that emotions have a certain range to them, and they really serve an adaptive function. And so today's talk, I will focus in particular on fear. And I think it's very intuitive to all of us that fear is very adaptive when kept at the right balance. Namely, if you're crossing like this good bridge where you can see that the ground is really far away, or when you're climbing, emotions help us to not take too much risk, yet to keep safe, but usually they still allow us to function in a certain range. And so at the level of homeostasis or balance of fear, it is totally adaptive. However, you may all know also that the balance can tick, and then, you know, this may result in different disorders, such as if there is too much avoidance or inhibition of action, this will result in anxiety disorders, or maybe into excessive risk-taking or the acceptance of otherwise very aversive consequences. So essentially, fear allows for flexible behavior, and in very simple terms, it is trying to establish a balance between avoidance and seeking that is at the best service for the individual's survival and also the survival of social groups. And that this balance is somehow difficult sometimes to keep is very well illustrated by the fact that anxiety disorders are really having a very high prevalence in the population. Depending on how you describe prevalence, up to 20% in a year, for instance, of the population are affected by anxiety disorders. And on the other hand, I would argue at least some parts of addiction disorders are also a disinhibition of maybe anxiety or avoidance behavior, of certain negative consequences. And the seeking here really does gain control of the individuum. And also these disorders are highly prevalent in the population, up to 4 or 5%. So that's why I kind of want to highlight in this talk an idea that we have in the lab, which is essentially that of homeostasis of emotions. So in general terms, homeostasis is the ability of a living system to maintain essential survival function in a condition of optimal functioning. And then emotion homeostasis could be something like the ability of an animal to maintain its emotion in an optimal range to serve survival. And so what do we need for an homeostatic model? Well, usually homeostasis is described by set points. So essentially a system that would know where is the adaptive range of a certain variable, such as an emotion, or as I will highlight today, of fear. Then there should be a certain regulator that usually maintains a balance. And in cases that weight on one or the other side of this balance is shifting, can initiate actions through effector systems that will rebalance the whole system. So very simply speaking, in the case of thirst, on these two sides would be the fluid intake and the fluid expenditure. And if you get too much expenditure, for instance, that you may need to drink. Now, does something like this also exist for emotions? We'll see today. And the proposal we're having is really trying to understand whether the insular cortex may be such an homeostatic regulator. On the one hand, the insular cortex is already known and has been described to serve bodily homeostasis, such as thirst or hunger, but also regulating arterial blood pressure and so on. And the insular cortex is also very much involved in emotions. So I want to just highlight very quickly to you which brain region I'm speaking about when I talk about the insular cortex. So here is an illustration of the position of the insular cortex in the human brain. Interestingly, it is a cortical region, but it cannot be seen from the surface of the brain unless you pull apart the overlying lobes. And so here in these cross sections of the brain, you can really see how it is invaginated at the level of the lateral sulcus. In the mouse brain, which is the model organism we are working in the lab, the insular cortex is located here on the lateral ventral side of the brain. And the most anterior part is here hidden behind the eye. So it's very hard to access brain structure, yet it's really occupying a rather large position. And the insular cortex is really interesting to us because it's really a hub of integration. It has access to interoceptive information from the gut, the heart, the lungs, and so on. It has access to exteroceptive information such as hearing, seeing, tasting, smelling. And it is also heavily connected to cognitive structures, such as, for instance, the prefrontal areas, the cingulate areas, the hippocampus. And we're in my lab really focusing and trying to understand how these different information routes are integrated in the insular cortex, which is, by the way, also very strongly interconnected with the amygdala, and what roles it may serve in emotion and bodily homeostasis. Another motivation to study the insular cortex is that it is really a hub affected across many very diverse psychiatric illnesses, as illustrated here in this meta-study. And many of those disorders that show functional but also connectivity alterations are disorders of emotion and mood, such as anxiety disorders, depression, addiction, bipolar disorder, and so on. So coming back a little bit to my introduction of fear being an adaptive function, the insular cortex has been shown to be hyperactive in physiological fear, so in healthy subjects. You can see it here alongside with the amygdala. But it is also across many anxiety disorders hyperactive, in contrast to prefrontal areas which are hypoactive. Furthermore, a very nice study already more than 10 years ago showed then that when patients suffer a lesion to the insular cortex, they abruptly stopped addictive behaviors such as cigarette smoking. And so there is really this role, a potential role for the insular cortex of modifying seemingly very diverse functions. And so the main questions I want to address today is one, does the insular cortex represent emotional set points? And two, does the insular cortex regulate fear? So to the first question, I would like to present you just very briefly some work done by a very talented student in my lab, Nadine, who performed 2 photon imaging in the insular cortex of freely moving mice. The data I'm going to present is already published, so I'm going to be rather fast and then come to the unpublished studies. So this is our approach of imaging the insular cortex. We essentially have a lateral moving objective, so the microscope rotates around the head of the animal so that we can image the insular cortex through a chronically implanted window here at the lateral surface of the brain. And we capture about the most posterior 2/3 of this structure because, as I said, the eye is occluding the most anterior parts here. And the first thing we did in this study was to try to understand which are the different stimuli and states that some of them had been hinted on by the literature are really represented in single neurons in the insular cortex. And what we found was not super surprising, but still nice to see. We found that the insula was activated by acute sensory stimulation, external sensory stimulation, such as tail shocks, which reflect also painful stimuli, but also tastes such as sucrose or quinine. And these stimuli were represented in very variously overlapping ways, so very multisensory. Furthermore, we also knew from the literature that we suspected the insular cortex would be activated by a change in bodily state, such as nausea, which we induced by the injection of lithium chloride. And what you can see is that overall, many neurons, about 3/4 of them, really statistically increase activity upon this bodily challenge. What we also found, and that was a little bit more surprising to us, was that the insular cortex, when animals were water deprived, was very much more active overall than animals where their thirst was quenched, suggesting that also this aversive or homeostatic disbalance was represented in the insular cortex. And so the question arose then whether also emotional states were encoded in the insular cortex, since the literature in humans at least suggested that fear or anxiety were encoded. And so what Nate did was a rather simple experiment. He performed fear conditioning in the head fixed condition, where he first habituated animals to tones. Then he performed fear conditioning where he paired the same tones to noxious tail shock. And there was a break of 60 minutes, and then he performed extinction learning. And I will only have time to show you some snippets of those data, focusing really on some times right after these different phases. These are always 5 minutes at the end of all those different phases, where no more sensory stimuli were delivered. The animals were actually remaining calm. However, what I tried to indicate here in gray is that obviously the fear or anxiety state of those animals was drastically different, as is also reflected in escape and freezing reactions. So after habituation, we would expect a low anxiety state that would rise through training, remain high by just giving the animals time to sit there, but then throughout extinction, we would expect a lowering of the anxiety. And what we saw in the insular cortex was that indeed we found that here there are 200 neurons from one animal, that overall the activity in those neurons really was strongly rising, remained high, and then only returned to baseline levels at extinction, which suggested to us that somehow this state of high anxiety was encoded here. And we made sure this is not only due to movement artifacts or arousal factors. It really seems to be somehow related to the state of anxiety. And we wanted to confirm this with a different technique. So this is where Alex Klein comes in. She's a very brave and talented student in my lab. And she performed electrophysiology recordings in the posterior insular cortex, again during fear conditioning. She recorded, in this case here, 25 single neurons identified throughout all sessions of the extinction paradigm, and then tried to see how were the reactivities of these neurons changing throughout the sessions. And what she could show is that she found that about 2/3 of the neurons were either activated or inhibited. So changed very drastically during this recall phase, which is characterized by higher levels of freezing, as you can see here. Again, suggesting that maybe something drastic changes during this periods of high anxiety in the insular cortex. And so what we did was really inspired by very elegant work by Yoav Livne from the Mark Anderman's lab. And what he did in a paper in Neuron last year was that he was interested in the insular cortex in the context of thirst, and to see whether the insular cortex encoded states of thirst, he concatenated ongoing activity. And so we did something a little bit similar, inspired by their work, where we concatenated equal amounts of ongoing activity in 100 millisecond bins from all the different phases of our fear conditioning paradigm. And we then performed principal component analysis, which you can see here. Every dot in here is essentially one of those time bins, and the lines are just in which temporal order they are here. And I plotted here the principal component 2 and 3, because this is where we thought the most interesting stuff is happening. And so what I now do is I color code for you the different phases of the fear conditioning. And what you can see is that towards the right, we have the habituation, which is a low fear state. Then the next temporally closed session would be the recall session, but it's much to the left. And in between, we have the extinction session, which we color-coded here in early and late. And this suggested to us that actually the principal component 2 might reflect in some way the state of anxiety. And another way to look at this was then to plot for each of these 100 millisecond time bins, which were not contaminated by freezing episodes, but we looked in the surround of these 100 millisecond time bins. What is the probability of freezing to occur? And when we color-coded then the freezing probability, what you can hopefully see is that we really find a mapping of this anxiety state in the insular cortical neuronal responses in this ongoing activity, which doesn't really reflect active fear expression. So what I hope I convinced you of is that in conclusion, we find that the insular cortex detects deviations from homeostatic and emotional set points. And this is achieved most likely through interoception and exteroception, although we do not really know how this is happening and will be a subject of future investigations. Now, the next question then would be, if the insular cortex is in a position to detect deviations from a set point, is it also an effector or a homeostatic regulator? Does it trigger actions to re-establish the set point? And so this is a project that Alex did during her PhD in my lab, where she used a classical fear conditioning paradigm. And what we were in particular interested in is the capacity of animals to extinguish their fear. So to relearn a previously formed association is not harmful anymore. So essentially, we do a very classical trick of habituating animals first to a tone representation in a certain context. Then we pair in the same context the same tone to foot shock, which makes the animals then learn that the tone is a fearful cue. During a memory test, we then ask, is the animal still afraid of this context and the fear cue? And then we put them through extinction learning, where in a different context, they are presented with this tone over and over again without negative consequences. And so they start to inhibit their fear. And during these tone representations, we inhibited the insular cortex. And the first thing Alex observed was in a first experiment that this treatment led to a facilitation of extinction learning. And that was significant, but it was rather subtle, and we did perform more extinction sessions. And essentially, after 2 extinction sessions, the animals were, both groups had extinguished. And so we thought this is rather weak. Can we kind of make it stronger by enhancing the strength and length of this foot shock so that the animals would exhibit more fear in the control condition, and maybe we would get a larger effect size. And what we actually observed was that, and we couldn't believe this for a long time, that this very small alterations, namely conditioning animals stronger, flipped the sign of this outcome entirely around. So now we find that the animals that were insular inhibited do have an impairment in fear extinction learning. And I put you here the performance index, which we calculate essentially as the decrease from the recall level to the last extinction tone presentation. And I hope what you can see is that this is enhanced in the low fear condition, but it's essentially impaired in the strong fear condition. We wanted to corroborate these findings because they were so paradoxical, and so we wanted to see whether within the same animals we could get the bidirectional effect, and that's why Alex performed consecutive fear conditioning, where she first conditioned animals to one set of tones in a weak conditioning paradigm, and then she performed strong fear conditioning. And I only show you here essentially would be more extinction sessions. So after extinction, after the fear conditioning week, we essentially extinguished all fear, and we waited several weeks so that when we performed the second set of fear conditioning with a strong stimulus, we didn't have much of a difference, as you can see here, between controls and halorhodopsin groups. But what we found is really confirming this idea that insular inhibition seems to make these effects of the state of the animal to matter. So this is plotted here again in this extinction performance index. And what I hope you can see is that the gray bars here are strong and weak fear conditioning in controls. And their performance is actually not significantly different. Meaning for a control animal, it doesn't matter how strongly it was conditioned, it will always extinguish well. While when we inhibit the insula, we disrupt this capacity. So the idea of state dependency I plotted here in a, I hope, more intuitive way. So what we did here is we plot on this axis for each animal the recall fear levels. And then we do see how well did they extinguish. And what you see here at the moment is the control animals, so when the insular cortex is intact. And what I hope you can appreciate is that this line is pretty flat. So irrespective of whether you enter fear extinction learning with very high fear levels or low fear levels, you do actually still extinguish very well. The same idea is plotted here, where I just plotted the extreme groups. And this is the recall freezing level, where you can see high fear and low fear animals extremely different. But then in extinction, they somehow merge onto a common denominator. They are exactly the same in control animals. Now, when we inhibit the insular cortex, we entirely disrupt this balance, and we make high fear animals being impaired and low fear animals being facilitated, as you can see here. Now, I think this is a very strong argument for arguing that the insular cortex somehow establishes homeostatic fear levels when it is intact, and what we do when we inhibit it is we disrupt this. But how could this be achieved? And so we came up with the model where this is what we observe in our data, right? High fear animals react this way and low fear animals react this way. But then how is this achieved in physiological normal conditions? Well, this argues that somehow high fear animals must have much less strong aversive teaching signals, because they don't extinguish so much of the fear, while low fear animals must have more strong. And so we wondered, can we see this in the activity of the insular cortex? And that's why Alex performed phybophotometry experiments. And so she did this throughout fear conditioning. And I show you the reactions to the soon to become CS+, so conditioned stimulus paired to a foot jog tones. And what you can see, the first two tone presentations during the fear conditioning do not elicit insular activation. But then throughout the conditioning, the animal learns and predicts that this cue will lead to an aversive foot jog. And what you can see is the insula starts reacting to it more and more. And then during extinction, we actually see that at the beginning of extinction, there is a net increase to the tone in the insular cortex and activity. And this then, through consecutive extinction learning, is inversed in such a way that at the end of a successful extinction, we actually do see almost an inhibition of the insular cortex with the presentation of the formerly CS+. And so in control animals, it's a bit stuck here. Okay, now I moved too fast. So what we see with this phybophotometry animals in control animals is that we see a correlation between the activity of the insular cortex in response to the tones throughout the different phases and the freezing that these tones evoke, essentially arguing that the more fear or the more harm predicting a cue is to the animal, the more the insular cortex will react to it. However, we saw that it makes a big difference to the insular cortex whether an animal enters with high or low fear into the extinction session. And so we just separated our animals into a group of high fear, as you can see here, or low fear animals. Their extinction performances were not different, as we had suspected from control animals, but they really, the high fear animals show a much stronger freezing decrease compared to the low fear animals, so to establish a homeostatic balance. Now, how is this achieved? Well, what we see then, if we look at the CS+ evoked responses on average in the insular cortex, we see that low fear animals actually activate their insular cortex at the presentation of the CS+, but high fear animals, very paradoxically, react with a lowering, so a decrease of the activity. And this was very, very surprising to us, because I just showed you that the predictive value of the CS+ should actually direct the signal to the top. So we had expected that high fear animals have higher responses, but this is not what we observed. So this is not only true in these groups or artificial subdivisions, this is actually true throughout. So here I just plotted for you the correlation between freezing during recall and insular reactivity. And as you can see, this is negatively correlated. So the more you freeze, the less you react in extinction. So This is in line, though, with our model that I put out earlier, namely that high fear animals have less aversive teaching signals and low fear animals have more aversive teaching signals. And our reactivity here is bidirectional. But how is this achieved? So we actually had a lot of misses and false guesses, but eventually I think we had the right guess. And this is to look at the insular cortex role in interoception. And it is known that the insular cortex, also called visceral insular cortex, receiving a lot of bodily feedback signals. And so what we looked at here is at heart rate. So here I already split the animals for you into low fear and high fear animals throughout habituation, fear conditioning, recall, and extinction. And I hope what you can appreciate is that the learning of the fear and the recall of the fear, but also then the extinction, leads to strong variations in heart rate, and they are mostly in sign negative. Now, this made us think that, and yes, the deviations are much stronger in high fear animals. So that made us think the real difference between high fear and low fear animals and what is really happening here is a lot of freezing reactions. And it was known in the literature that freezing actually goes along with a decrease in heart rate, a strong drop in heart rate. And this is what I show you here in an experiment where we combined heart rate measurements and photophotometry in the insular cortex. The first thing we see is heart rate drops at freezing onset, and concomitantly also insular activity drops, most likely by the detection of this heart rate drop. And this is what we wanted to know. Is this insular drop in activity linked to the heart rate? And so we didn't want to manipulate the heart directly, but we turned to the vagus nerve, which is the main sensory afferent of the brain from the body. And it is known to also carry interoceptive information to the insular cortex. And to manipulate the vagus nerve, we actually turned to vagus nerve stimulation, which is a clinically applied technique to override essentially the tone of the vagus nerve. And this is already approved for the clinic to treat epilepsy, but it's also successfully applied in depression, and it's tested for anxiety disorders. So we developed a vagus nerve stimulation paradigm for mice in order to override the communication from the heart with the brain. And so first, to show you this, so we here combined vagus nerve stimulation with photophotometry in the insular cortex. And the first thing we wanted to make sure is that indeed, if we stimulate the vagus nerve, we see a change of activity in the insula, and this is shown here. So the green shaded area is the vagus nerve stimulation, and the Z-score here is the activity of the insular cortex measured by photophotometry. You can see this is nicely activated. So really arguing that what we are doing here is we are affecting activity in the insula through the periphery. And then we applied vagus nerve stimulation in our fear conditioning paradigm in a similar way as we had applied inhibition, namely during the tone presentations throughout extinction learning. And the first thing we saw is that here in the sham, or this is actually not a sham, this is the same animals, but outside of VNS, we see what we would expect, namely a drop of insular activity at the freezing onset. And the first thing I want to show here is that with VNS, we override this drop. So we do not see this bodily feedback signal affecting the insular cortex in the same way anymore. And so the next thing we checked is how does the insular cortex then react to the tones when we couple this with the vagus nerve stimulation. And what it actually does, it inverts the correlation that I showed you before. Namely now, high freezing animals have a higher CS plus evoked activity in the insular cortex and low fear animals. So somehow there seems an integration between these two signals in the insula, namely the predictive value and the bodily signals. And when we stimulate the vagus nerve, we override this. And this then results also in an inversion of the sign of the teaching signals, because now we report that in high fear animals, we see an increased activity upon the CS plus, and in low fear animals, we see only a minor increase, maybe even an inversion. So if I put these data side by side, I hope you can appreciate that in control animals, we saw that low fear animals had an increase of the insular activity to the CS plus, but high fear animals had a drop. Now upon VNS, we invert the sign of these teaching signals. And then finally, what we obviously wanted to check is what influence does that have on the ability of the animals to learn fear extinction? And what I can show you here is that it actually has the exact same effect as inhibiting the insular cortex itself. Again, what you see, I divided the animals here into high fear and low fear groups during recall. The controls here in gray then reach a homeostatic level and of adaptive fear extinction, and the VNS-treated animals react when they are high fear with an impairment and when they are low fear with a facilitation of extinction. And this is then resulting in this state-dependent extinction performance, which is essentially the balance that we observe in the controls is disrupted in the exact same way that we had seen in PIC or insular cortex inhibited animals. So we disrupt the balance of extinction performance. So I just want to put this all together for you. So I hope what I have showed you, this is our working model at the moment, that the insular cortex is in a position to detect homeostatic set points, in this case, the levels of fear which are adaptive. And when these fear levels become not adaptive anymore, it actually tries to reset the balance. And this seems to be achieved by reading out signals that are produced by the expression of fear itself. So freezing in our case, which translates into somatic changes, namely heart rate decreases, which go alongside with a decrease in the activity of the insular cortex, which in turn is then transmitted via the vagus nerve to the insula. And so we currently believe that these bidirectional teaching signals are somehow fed into a fear and extinction network that many of you know about and have worked extensively on to really gate extinction performance to homeostatic levels. So we propose a role for the insular cortex in emotion homeostasis by, as I said, predicting from cues, but also bodily signals, the integrated state of the animal and providing teaching signals to revert to the set point. And what we do not know is what the effector systems of this are, and we have some suspicions. So we know that this part of the insular cortex is very, very strongly connected to the central amygdala, actually also the bed nucleus of the striatum terminalis. It's also connected to the basolateral amygdala, the accumbens, and the prefrontal cortex. So there are a lot of possibilities of an intricate network that could serve those functions. And so with this, I already arrive at the end of my today's talk. So I would like to thank my team very much. They're really an awesome crowd to work with. So they're all listed here. Today, I spoke mostly about Alexandra Klein's work, who's an awesome student in my lab, and she will be going on for postdoc very soon. And Nadine, who performed all the 2-photon imaging experiments. And here are my funding sources. With this, I would like to thank you all very much for your attention.
Excellent. Thank you so much, Nadine. Can everybody hear me? Good, I'm back on. So I apologize for some technical difficulties we had earlier. I'm Eric Nestler from New York City. David, you want to start a few questions? We do have some in the chat.
Yes, I will go ahead.
And I just remind people, if you do have questions, add them to ask a question at the bottom of your screen. Go ahead, David.
Okay, so I'm going to go in the order of basically upvotes here. So the first question is, thanks for the interesting talk. We generally refer to homeostasis in biology for biological variables that can be measured and that are unidimensional, so like pH, temperature, glycemia. But in the case of emotion, how would you define a homeostatic reference, especially since we tend to measure emotions through various indirect measures, often related to the autonomous system?
You want the long or the brief answer? The brief answer is I do not know. I go by the data. I think it's an interesting question, but I think that the idea of homeostasis still kind of fits. And we just, for emotions, we're really, really at the origin of understanding what they really are and how they can be described. It will be multifactorial, I'm sure. It will not just be one variable, but I mean, I think that it is maybe also simplistic for some bodily homeostatic. So I think thirst is also very much modulated by satiety, by temperature, and so on. So there's also prediction playing into play of how the current environment will affect the fluid need. And so I don't think it's that easy for most biological problems. And I think for emotions, obviously, it's maybe the most complicated.
Nadine, another question is the synaptic connections between the insular cortex and the vagus. Do you know how many synapses are interposed between the two?
Yeah, there are at least 3 or 4. It's not very direct, yet it is still very consistent that the vagus nerve is supposed to arrive in the nucleus tractus solitarius, and then it goes through several stations, such as the PBN, and it's really somehow a project that I would love to do some multi-synaptic tracing, and there have been very, very nice studies from the gut into the brain, also from the lungs into the brain, but I don't know which are the relevant. I mean, we here describe a relevance for heart rate, but is this the only one? I do not know. It may be a little bit more. So yeah, this is really a dream project I would like to do in the future to see. But even with tracing, you don't know whether this is a way that's taken. So there is a lot to do to understand better how this is done.
I'd like to ask you a question. There are some humans with vagus nerve stimulators for epilepsy primarily, or in some cases for depression. I wonder if one can do a study to see whether frequencies of vagus nerve stimulation in humans are affecting activity in insular cortex with brain imaging.
Yeah, that actually has been done. So it activates. I don't know whether a very nice titration study has been done. I'm not so sure, but that the vagus nerve stimulation alters insular activity was actually how we got the idea of the vagus nerve. Yeah.
Oh, that's awesome. Yeah, great. Go ahead, David.
Hi, so another question. In the early slides, it seems about a third of insulin neurons were activated by fear while a third were inhibited. Is it possible that the high fear, low fear opposite response is associated with these two populations? And I would like to tack on, are the molecular profiles of these neurons different? Do you think it could potentially be inhibitory neurons versus excitatory within those synapses?
Yeah, so we are pretty sure they are not inhibitory excitatory. I don't know whether they could have different projection profiles and then hopefully also molecular profiles. We do not know that. I don't think the high fear, low fear is divided in that way. I do not think that, because we have shown in our consecutive fear conditioning experiment that there is some sort of trait involved, so that high fear animals tend to be high fear both upon weak and strong fear conditioning, but it's not 100% penetrant. you can also be a low reactor and become a strong reactor. There must be other factors playing into this. And since every animal, I think, has the capacity, as we have shown in the separate experiments, to, upon different exposures, be more reacting like a low fear or like a high fear, I do not really think that it's a circuitry.
One of the other questions is the degree to which you can generalize emotion from fear. In other words, what role do you think the insular cortex might be playing in other emotional states?
Yeah, so from the other work that I didn't talk about today, we are pretty certain that the insula plays also a role in more appetitive emotional states, but also in disgust-like states. I don't know whether this, I mean, my kind of naive thinking is that this kind of idea of being a detector of something goes out of range is pretty, maybe pretty generalizable, but has to be shown. But we just know that the insular cortex is not just reacting to one emotion. It's not just serving fear or anxiety.
Mm-hmm.
And then I think we have time for a couple more, but another interesting question was, fantastic talk. I thought that VNS was supposed to reduce anxiety and psychiatric disorder, but this work ought to predict that high fear individuals should get worse following VNS, or have I misunderstood?
Now, whoever asked has understood very well. Now, I'm not a psychiatrist, right? So I don't know whether I can project my data onto the human. And I think there is definitely space for titrating the stimulation paradigm to make it fit to what is beneficial. But yes, our data suggests that there could be a potential for harm. And as far as I know, many of the VNS studies carried out have treated fear in healthy subjects. But there's also a lot of work from the rat, where there has been a whole range of studies where vagus nerve stimulation was beneficial. And I don't really know, maybe it's a difference in protocols or in just pooling data. We do see the facilitation effect. We just also see, if the animals are really in high fear, that it is actually counteracting the extinction. And so it's at least something I would say is worth looking for in human patients, because you also have a lot of dropouts in those studies, right? So people sometimes start a test and then they may drop out, and it's really hard to track what it's due to.
One of the other questions, Nadine, is asking for about the role of the insular cortex in depression.
Mm-hmm. Yeah, I would love to know. I think the only answer I have, we will definitely investigate this. Yeah.
Let me see here.
And then we still have some time. There was another question from Alejandro Pacheco, who is commenting on the single cell calcium imaging experiments that you showed us, and whether you know whether the same insular neurons are responding to the different types of signals that you've studied.
Yes, so I would refer him, I think, or them to the paper is published. We haven't exhaustively analyzed the data yet to make sense of the overlap in different responses, but there is a very, very strong overlaps in very mixed ways. So we have overlap in single neurons of all kinds of combinations of the different stimuli used. And so I would just refer to the Nature Neuroscience paper to look it up, and then hopefully stay tuned for until we make sense of it.
Mm-hmm.
David, do you see another one that you'd like to ask?
Let's see.
I mean, here's an interesting question. I'm just going to read it because it's a long one, Nadine. So thank you for an amazing talk. In the introduction, I found it interesting to put anxiety and addiction at two ends of a spectrum. Do you think when insular damage helped people quit smoking, was it because of decreased anxiety or decreased risk-taking? In your experiment with thirsty mice, you showed that satiating a craving decreases insular activity. Do you think smoking could be similar for an addict?
Yeah, I kind of was prepared for the question, and I still don't have a super good. I think a very hand-waving answer could be that psychiatric illnesses are really, I think, for me, I'm still also a layman in this because I'm not a psychiatrist by training, but they are very comorbid, right? So you have comorbidities between depression and addiction, anxiety and addiction, and the insula seems to be a hub in all this. And so I think it's really difficult. And I think maybe, for me at least, the best way trying to understand these cross correlations would be by doing it in animal work in a controlled way. And this is a very nice idea, right? Can you influence the different variables by substances of abuse or by suppressing craving? Yeah, that's a very nice idea, but we haven't done it yet.
Mm-hmm. Yeah.
I think the last question we have here too is, can the set point shift be mediated by plasticity within the insula itself?
Yes, we don't know. I would say yes. So I talk about these ideas a lot with Mark Anderman, who is more on the bodily homeostasis side at the moment, at least. And I think there are some interesting psychiatric illnesses, such as anorexia, for instance, where maybe the set point shifts, where the desired state is being hungry. And maybe, in some other psychiatric illnesses, it's similar. I mean, anxiety-prone subjects, they try often to cope by being prepared for everything, right? So they also shift their set point somehow to being always alerted. Trying to prevent harm from arriving. And so, yeah, definitely a very interesting question also.
And because of the technical difficulties, Nadine, I missed part of David's introduction, but you're in Martinsried, Germany, which is west of Munich. Tell us a bit about your Max Planck Institute.
Yes. Okay, so we are Institute of Neurobiology. So there's a lot of circuit neuroscience going on here across different species, so flies, zebrafish, mice, and there's a lot of imaging from whole brain imaging in zebrafish to what we are doing, or others are doing, to photon imaging, also to photon miniscope imaging. We are now have a new group leader is doing functional ultrasound whole brain imaging. So it's a very nice place. It's near to the Alps. It's in Europe, and it's a very interesting and vibrant institute. Yeah. So if anyone is interested, just drop me a line. I can highly recommend it.
Fantastic. Thank you so much for a wonderful talk today. I really enjoyed it a great deal. And thank you everyone for joining us. We do want to note that there is a lecture next week from Gavin McNally. It's going to be at 6 p.m., I believe, in European time. Daylight savings time in the United States is not synced up with the rest of the world, so we have some confusion about the hours. But please stay tuned for all the announcement on the web. And thank you for joining us. Nadine, David, thank you.
Thank you.
Bye-bye.
Bye.